I should like to tell you that I have been working on the experiments shown in this film in the Nebular Mitosis Laboratory in the Department of Planetary Futures here at The Ohio State University. Realizing the urgent need for a Planet B, our research is generously supported through the Institute of World Forming, the leading international planetary research agency.
What if humans could harness the wisdom, science, and magic of plants to form new planets? That is the question we are working to answer here in the laboratory. In our hybrid program, ACSE, for Allium cepa stella erratica, or onion planet, our goal is to contribute to understanding how to continue life as humans know it by ensuring there is a Planet B. We are grateful to the scientists who work to invent and improve the new technologies we utilize. We strive to use ingenuity and creativity in developing innovative approaches when interacting with materials. The methods you are about to see aim to understand the process of common onion propagation to form a new planet. This is done through a series of experiments which analogize biological and astronomical processes such as mitosis and the nebular hypothesis. In a tiny sample of an onion root tip, several stages of cell division can be seen all at once. This rapid growth makes Allium cepa an ideal planetary candidate. We are confident that Allium cepa will bring the genetic information that makes its perfectly spherical shape and rapid growth with it as its scale increases greatly over time and it begins to orbit the solar system. Our laboratory has 4 main growth chambers, each engineered to uniquely support the appropriate phases of biological planet formation, which are rooting, bulbing, bolting, and fusion. As you can imagine, technique is everything. Growth Chamber 1 is a hydroponic incubator for pearl onions and supports both rooting and bolting onions before they are entered into the fusion process. Growth Chamber 2 supports full-size Allium cepa during their rooting stage. They are removed from the incubator to receive root cleanses and water replenishment. Growth Chamber 3 supports Allium cepa once they have reached the bolting stage, which begins when the green leaves or bolts of the onion begin protruding out of the sphere. Both plants and planets rely on the sun. In order to achieve the appropriate spectrum of light, special artificial conditions must be created. Grow lamps are utilized to simulate the sun and encourage optimal bolting. Through the accumulation fusion technique, many onions may be combined in protoplanet formation, as seen here in Growth Chamber 4, where nucleation occurs. This protoplanet is currently being weaned from the Earth's atmosphere to prepare it for its propulsion into outer space. The process of fusion is one of the most extensive processes in the laboratory. Scientists have learned that planets are formed from a solar nebula, residual dust and gas left from the sun's formation. The accumulation or clumping of material over time is crucial. Specific minerals are intertwined in the roots and bolts to simulate a dense center for planet formation to begin. We are currently investigating the optimal time to begin the accumulation and fusion processes. Failed formation attempts are preserved for documentation and educational purposes. One form of quality control in our laboratory practice is to section and print representative Allium cepa. Analytic heat prints show us the development of the interior of each onion analyzed. The prints guide us in understanding the optimal timing for accumulation and fusion. They also give us information about the onions that fail to produce roots and bolts. You may still be asking, why onions? The obvious connection is the impeccable shape, which indicates that an onion is the perfect seed for a planet. The spherical nature is embedded in the onion's genetic code. If this perfectly round sphere has the capacity to create such astounding roots and tips so rapidly, It may also hold the key to unlocking the ability for humans to propagate the much-anticipated Planet P. ACE was a hybrid program in the Nebular Mitosis Laboratory of the Department of Planetary Futures. Housed within the Urban Art Space in Columbus, Ohio, ACE was open to the public. There were 2 ways for the public to experience the laboratory. The first was during set lab hours, which were Monday through Friday from 11 to 2. During this time, I was in the lab tending to allium cepa and machinery, performing experiments and documenting findings. In my presence, Growth Chamber 3, the central breathing inflated incubator, was in full operation. Daily tours were offered, which included the interior of Growth Chamber 3. Conversely, during off hours, the laboratory was open to be viewed, but tours were not possible, as Growth Chamber 3 was deflated. Though allium cepa were still within and could be seen somewhat through the milky translucent polyethylene. A microscopy station was available for viewers to look into, mounted with a slide containing 3 allium cepa root tips, which displayed several stages of mitosis simultaneously. The lab notebook was left open on the desk so that viewers could page through to learn more about the lab operations and the experiments that were underway around them. Additionally, lab clipboards full of notes hung on the wall next to a monitor which played the introductory video about the ACE laboratory you saw in the first portion of this presentation. The exhibition, and therefore laboratory, was set to close on March 14, 2020. But this closure was accelerated the week before as a global pandemic, COVID-19, which had been sweeping across the world in previous weeks, was now making its way across the United States. Accordingly, on March 10th, the process of preserving all live planetary propagules began. Each bulb allium cepa with bolts and roots was moved from Growth Chamber 3 to a 32-ounce glass specimen jar. The identification sticker was transferred from the bulb allium cepa beaker to their new jar. The jars containing planetary propagules were then filled with white vinegar for preservation and tightly capped with a plastic lid. In conclusion, the ACE laboratory ran for a concentrated and short duration, in which time principal experiments were conducted to advance the understanding and functionality of the cross-disciplinary relationship of astrobiological methodologies. From the nascent stage of protoplanet propagation, which included 3 main growth chambers in which both bulb and pearl allium cepa were nurtured and tended through the rooting, bolting, and bulbing stages. Through detailed observation, ACE found the optimal time and methodology for fusion in which 4 protoplanets were created. 3 of those planets were subsequently preserved. 1 vital planetesimal, Primus protoplanet, is currently in the process of weaning and preparing for space propulsion. These experiments and resulting findings pioneered new processes to aid in the future of human colonization and habitation in outer space.